Okay, so this video is going to look at the different biological molecules we've got in our body, a little bit about their structure, and then goes into an important type of molecule called enzymes, just what we need to know on a GCSE basis. Well, one of the first molecules we've got are the proteins. Proteins are important for different reasons. We need them for building the structure of our muscles. We also need to make a lot of the chemicals, or nearly all of the chemicals in our body out of them. So things like hormones, neurotransmitter that's used in our nervous system, and the molecule I'll be talking about soon, the enzymes, they're all proteins. So without it, really, we wouldn't be existing. If you want to go further from there, well, the eye colour, hair colour, skin strength, it's all down to proteins. We've also got carbohydrates as well. And in biology, there's starch and glycogen. Starch is mainly found in plants because plants use starch to store things, whereas we use glycogen. And our carbohydrates are very much for energy. Plants also use it for things like making cell walls. You've probably heard of cellulose in one of the videos. In us, it tends to be used, as I said, for energy. In plants, it's used for energy, but also for certain structures as well. And then the third type of molecule are the lipids. Lipids can be used to make our nervous system, or they can be used for insulation. Think of a walrus and how it uses a lot of fat to keep itself warm in the cold ecosystem it comes from. It can be used for protection. We've got a nice little fatty coating on different organs, and it's also very good for storing energy. We actually can classify lipids as two different things, fats and oils. Fats are the solid type that we tend to have, whether we are having animals, whereas oils tend to come from plants. So what are these molecules actually made of? Well, you can see here that this protein is made up of a big long chain of little shapes, and those shapes represent molecules called amino acids. So proteins are made of a big long chain of amino acids. So when we eat in, we actually absorb amino acids, or at least we break down proteins in our diet to get amino acids so that we can make our own proteins. And just like with the proteins, which is made up of this chain, carbohydrates are also made of a chain, but in this case it's made of sugars. One particular one you'll have to know about is glucose, that we do when we talk about respiration, but that's another topic. Fats are a little bit different, or the lipids are, I should say, are a little bit different because they're not a chain. Instead, they're made of two different types, glycerol and fatty acids. So the most common type, what's known as a triglyceride, it's got three fatty acids represented by those zigzag structures, and a glycerol represented by that yellow structure. So we've got proteins that are made of amino acids, carbohydrates such as starch and glycogen that are made of sugars such as glucose, and lipids that are made of fatty acids and glycerol. We can actually test for these different molecules, and there's two that you have to know. The one up above we use in a substance called the test called the Benedict's test, and the substance will be blue at the start, but if we add glucose, that's the sugar from the carbohydrates, it will gradually turn to a red colour, depending on how much sugar is in the thing we add. Now, if you ever ask for this in an exam, we just say that it turns from blue to brick red. But it's the Benedict's test, testing for sugars, and it'll go blue to brick red if the test is positive. The one down below is the iodine test. Now, if you remember from one of the other videos, we said that potatoes contain a lot of starch because plants use them for storage. So when we add iodine to potato, because it's a test for starch, we'd expect to see a colour change. And indeed, this picture is showing that. The iodine in the bottle is a brown yellow colour, whereas a lot of it tends to look like a dark brown, like in this picture. But it'll turn black when there's starch present, and you can clearly see that on the potato, this iodine is turning black. So we've got the Benedict's test for glucose, which goes from blue to brick red, and the iodine test for starch that goes from a browny yellow to a black. Our last thing to look at are special molecules called enzymes. Now they're what we call biological catalysts. Biological meaning that they're in living organisms, and catalyst meaning that it's a chemical which speeds up a reaction. So these are proteins. So you've got lots of different types of enzymes in your body which speeds up the different reactions, and that's essential because otherwise they'd be too slow for us to actually do anything. So for instance, respiration is a crucial reaction that produces energy, and if it was really slow as it would occur if there was no enzymes, we just wouldn't get our energy to move, to grow, to do anything. Whereas because an enzyme is used to catalyze it, it's used as a biological catalyst, it speeds up respiration to get us enough energy. Now the important part of an enzyme is called the active site, and we say that the active site is complementary to the substrate. Now what am I actually talking about here? A substrate is the molecule that the enzyme is trying to help react. So it needs to hold on to that substrate somehow, so that maybe another molecule can come and react with it, or it puts a strain on the molecule so it's more likely to break apart. Whatever reaction it's doing, it has to hold this substrate, and the active site is specific to that shape of the substrate. The substrate can only lock into that active site. So you need enzymes for different types of molecules. If I was making a protein, then I'd need an enzyme with an active site that fits amino acids. If I was making a carbohydrate, I couldn't use that one because the substrate, the glucose, wouldn't match the active site. It wouldn't be complementary to it. So as a result, I'd need a different enzyme, one that helps bind glucose molecules together to create the carbohydrate. Now you've got to remember that the enzyme isn't taking part directly in this reaction. It's not changing and reacting with the molecules, it's helping them to react. So what's going to happen is when the substrate changes and just moves away, the enzyme can be used again. Now enzymes are affected by two factors. One of them is temperature. The trouble with temperature is that when it's cold, the enzyme doesn't have enough energy and it can't move around much. This means it won't collide with the substrates and it won't bind them to their active site, so the reaction doesn't occur. But what's going to happen is as the temperature increases, there's more energy from the heat, and so the enzyme can work faster. There's more collisions between the substrate and active site until it gets to the best point, what we call the optimum temperature. Now above this, it starts to damage the enzyme and the shape of the active site starts to change so that it no longer matches the substrate. So if you go to warm temperatures, then the actual rate of an enzyme reaction slows down because the enzyme is changing shape. It's what we call denaturing. A similar sort of thing happens if a pH is too high or too low. If a solution is very acidic or very alkaline, think of your stomach acid or maybe hydrochloric acid from an laboratory. Those things would also alter the shape of the enzyme's active site, and so the reaction would, the enzyme would denature and the reaction would slow down. So let's have a look at some questions then. What are the three types of molecules in the body? Spelling mistake there, sorry. Excluding DNA. Name the molecules that these are made of. What's the test for glucose and what colour does it change to? What test do we use for starch and what colour does it change to? What is an enzyme? What part of an enzyme is complementary to the substrate? And how does temperature affect enzymes? And what's the other factor that affects enzymes? So pause the video and have a try of those questions. Okay, so the answers. The three types of molecules that we said: carbohydrates, used for energy and to a degree storage of energy; proteins, which are used for growth and a lot of important chemicals such as enzymes in the body; and lipids, used for storing energy, insulation, protection, and so on. Name the molecules these are made of. Well, remember, in that chain, proteins are made of amino acids, carbohydrates are made of sugars, such as glucose, and lipids aren't made of a chain, they're simply made of fatty acids and glycerol, usually 3 fatty acids and a glycerol. What's the test for glucose? Well, it's Benedict's test and it goes from blue to brick red. Test for starch is iodine going from brown to black. What's an enzyme? Proper way to say it is a biological catalyst. It is a protein, but we refer to it as a biological catalyst. What part of the enzyme is complementary to the substrate? That's saying what part of the enzyme matches the shape of the substrate. That's the active site that the substrate will bind to and lock into. How does temperature affect enzymes? Well, as the temperature is increasing, the enzyme's got more energy, so it speeds up. It gets to its best temperature, called the optimum temperature, and then as it gets too high, it damages the enzyme, denaturing it, and the enzyme slows down. And what's the other factor that affects enzymes? It's pH, acidity and alkalinity.